Alright guys, this is a recording of chapter 5, your next exam, the dynamic cell lecture outline. That's what we're going to look at. So some of the key concepts we're going to look at over here is potential energy, kinetic energy. Those are two basic forms of energy, although there are other forms of energy as well, but we're going to look at potential energy, which is stored energy. Um, Stored energy is energy that is stored because we are not in motion. Kinetic energy, on the other hand, is energy of motion. That means whatever is not stored, but that means we're in motion. So, for example, um, someone like a bicycle going down a hill, a ball rolling down the hill, or you throwing a football, or even us walking. That's energy of motion. Now, anything that's stored, such as energy that's found in food, is considered potential energy. Um, Something like just sitting there, not moving, is considered potential energy. The energy of a diver standing on a diving board, which you see, they're showing you figure 5.1 on page 80, the difference between kinetic energy and potential energy. But kinetic energy is energy of motion. You see the young lady walking up the steps to get to the pool. Um, for example, um, she's just standing there in the middle. That's potential energy. The stored energy of position or configuration. However, when she dives off the diving board, that potential energy is converted to kinetic energy because she's now in motion. Okay, so anything that's not in motion, a glucose molecule is considered a type of potential energy. Energy stored in foods is a type of potential energy. And the lady just standing at the top of the diving board and not moving is a type of potential energy as well, which is what you see here. Okay, measuring energy. Okay, the laws of thermodynamics are very important. You do need to be familiar with those as well. Um, so, um, the first law, okay, there are two laws. The first law is conservation of energy. Energy cannot be created or destroyed, but it can be changed from one form to another. What that simply means, kinetic, as we just looked at, potential energy can become kinetic energy. That's being changed from one form to another. Also, another good example would be um, the electricity, the light bulb, when you plug your lamp into the outlet, of course, the electricity is going to be transformed into light energy, which is going to give light to your room. Those are just some simple forms of how energy can be changed from one form um, to another. Okay. Second law, energy cannot be changed from one form to another because if it does, well, it can be, you have to have some energy that's going to be lost. Heat is the least usable form of energy. In your textbook, also associated with the second law of thermodynamics, which is on page 81, um, entropy is associated with this. It's going from a state of order or to a state of disorder. Page 81, the second law of thermodynamics tells us energy cannot be changed from one form to another without a loss of usable energy. Many forms of energy are usable, such as the energy of the sun and food and ATP. Um, so that's some examples, but it can be converted from one form to another. Um, energy Every energy transformation leads to an increase in the amount of disorganization or disorder. The term entropy refers to the relative amount of disorganization. Okay, only way to bring about or maintain order is to add more energy to a system. to take an example from your own experience, a tidy room is more organized and less stable, okay, um, than a messy room, which is disorganized and more stable, okay. So messy room, you can go from an organized state to a very disorganized state, which is entropy, that is actually associated with the second law of thermodynamics, and as stated, as stated, the disorganized room is less stable. And here it is here, relative amount of disorganization. Okay, I don't think that's on your test, but just wanted to bring it to your attention. Cells and entropy. This one right here, when you have an unequal distribution of hydrogen ions, you have more hydrogen ions outside of your cell as opposed to inside. This is going to be more organized and, of course, less stable because it has more potential energy and it's going to be less entropy. However, when the hydrogen ions start moving and you have an equal distribution on both sides, you have 4 on your right side and 4 on your left side, it's going to be a little bit less organized. There's going to be less potential energy. This one is going to be more stable because you have an equal distribution of hydrogen ions, and of course you're going to have more entropy. Adenosine triphosphate. ATP, guys, is the energy currency of our cells. ATP is what allows us to carry out all of our daily activity because it's called energy, adenosine triphosphate. Of course, this particular molecule is a nucleotide. It does have the most potential energy to perform the work for all of our cells. It doesn't have more kinetic energy, but it does have the more potential energy in order for the work in our cells to be carried out. So, for example, that would be ATP has to be broken down to ADP in a phosphate group in order for us to get this energy. So it's going to lose a phosphate group and it becomes what? Adenosine diphosphate. It's continuously recycled in regeneration, which is why it's called the energy currency of your cells. As stated, ATP, guys, it has the most potential energy to perform work for nearly all of our cell activities. And this is what it looks like. It's a cycle. Okay, you can see it being broken down to ADP and a phosphate group, and it's a coupling reaction. Okay. This is considered a coupled reaction as well. Here, this picture is in your textbook on page. Page 84, figure 5.5. So here, this is a really good example of a coupling reaction that's needed for your muscles to contract. So if you look at figure 5.5, it shows how ATP, if you look at the lady lifting the dumbbells, you have 2 proteins that's responsible for your muscles to contract in contracting, actin and myosin. ATP, of course, is bound to the myosin head. Okay, so during ATP is going to be broken down. If you look at this portion here, ATP is being broken down to that ADP in the phosphate group in order for the myosin head to actually bind to the actin. Okay, so the myosin filaments are going to pull the actin filaments to the center of the cell and the muscle shortens. So this is what's happening right here. So now she's doing a bicep curl. The myosin head has to attach to the active site on the actin filament in order for the muscle to shorten. So first, myosin has to combine with ATP, and only then does ATP break down to ADP in a phosphate group, and the release of ADP and the phosphate causes myosin to change shape and form the actin. So this is called a coupled reaction. Muscle contraction occurs only when it is coupled to ATP breakdown. Myosin combines with ATP prior to its breakdown. Release of ADP by plus the phosphate causes myosin to change position and pull on the actin filament. So muscle contraction um, when it's coupled occurs when it's coupled with ATP. This is called a coupling reaction. The flow of energy. Energy flows in one direction. Of course, it flows from the sunlight to the. This is not going to be on your test, but there's a one-way flow of energy. It's solar energy used to convert water and carbon dioxide into your carbohydrates, which is your food for plants and your organisms. Cellular respiration is a breakdown of your carbohydrates, particularly glucose, in order to build and release energy in the form of ATP. All right, so humans and energy. Yeah, how do we get our energy? Okay, we inhale oxygen, we eat the plants. The plants make their carbohydrates from the solar energy or from the sun. And we're considered animals because remember, we're part of that kingdom of animals. We eat what? The plants. So energy-rich foods allow us to produce the ATP required to maintain our bodies and to carry on our day-to-day activities. Okay. Enzymatic action. Okay, so we're getting ready to talk about enzymes over here, but I want to start another presentation for, well, I guess there's not a whole lot I have on your test about an enzyme, but okay, this is the part that you need to understand. Enzymes have what you call a substrate. If you look at the figure, this figure here, I don't know what page it's on in your textbook, the substrate has to bind to an active site. Okay, another name source for a substrate is a reactant. So the substrate binds to the active site here, and then you're going to have a chemical reaction that's going to occur. So enzymes, remember, are receptor proteins, and they're responsible for breaking down chemical messengers and things of that sort. So what's going to happen is going to break the substrate down right here, and it's going to create two products, okay? May facilitate breakdown or synthesis reactions, but you have to have a substrate, okay, in order for that to occur. So remember for your test that the substrate has to bind to the active site in order for a chemical reaction to occur. In order for the chemical reaction to occur. So the enzyme's active site. This is on page 86 of your textbook. The active site must accommodate the substrate. The substrate, okay? It's like a lock and key. It's called the induced fit model. So when the substrate binds to the active site of the enzyme is going to undergo a slight shape change to accommodate the substrate. When that happens, the reaction is going to occur. Okay, and once the reaction occurs, it's going to produce the product, and the active site is going to go back or resumes its shape. So the substrate, let's say, what would be a substrate? Enzyme would be, let's say, lactase. Most enzymes end in ASE. Lactose, of course, will be our substrate that has to bind to the enzyme lactase in order for lactase to break down lactose, so people won't be lactose intolerant. That's a really good example. Feedback inhibition, I don't think is on your exam, so moving on. This is what a feedback inhibition looks like. Of course, you're going to have your enzyme. You have six enzymes here. Well, actually, four. If you have four enzymes, the first substrate is going to bind to the active site on enzyme 1. Okay, so when that substrate binds to enzyme 1, you're going to create a pathway, and it's going to produce product. That product is going to bind to enzyme 2, the active site of enzyme 2, so forth and so on, and that's when you're going to have a feedback inhibition. So, a feedback inhibition right here, when enough of the product has been created and the body doesn't need any more, what's going to happen? That product is going to feed back and it's going to bind to the active site on the original enzyme, change its shape, and of course you can't produce any more product because the shape has been changed. So that's 16 minutes of that. I'm going to stop this one right here.